Okay, very good afternoon, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal, convener of this CPGET 2023. So we will going to sum up within 2 days. So in this regard, we would like to invite the very huge experienced personality in the botany fraternity. None of the other than Dr. B. Rukmini Devi Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Narsapr, Medak District. Madam, please welcome and share your screen and start your session, madam. I have given to you sharing option.
Okay, sir.
Okay,
Thank
madam.
you,
Please
sir.
welcome.
Thank you.
Madam, please welcome.
Thank you, sir. Good afternoon, everyone. Today we will discuss about sexual reproduction of Penicillium. In yesterday's class, we have discussed about Penicillium vegetative morphology, its systematic position, vegetative reproduction, and asexual reproduction. In today's class, we are going to discuss about sexual reproduction process in a detailed way. Then the economic importance of Penicillium species we will cover in today's class. Yes. Penicillium species reproduced by 3 methods actually. One is vegetative method, and another one is asexual method, and the third one is a sexual method. Actually, sexual reproduction or sexual stage is not reported in all the Penicillium species. Only few Penicillium species showing this sexual stage. This sexual stage is also called as perfect stage. So the species of Penicillium which are exhibiting this sexual stage those are called as perfect fungi. Only few species of Penicillium showing this sexual stage. Remaining reproduced only by asexual and vegetative means. So, in what type of species this sexual reproduction is taking place, we are discussing. One is Penicillium vermiculatum. In that species, sexual reproduction process will take place. And another species, Penicillium glaucum. In this also, sexual stage was reported. So, only few of the Penicillium species showing this sexual stage. So, in this Penicillium vermiculatum sexual reproduction process, we are discussing in a detailed manner in today's class. So, here from the well-developed mycelium, sex organs will develop. So, here the Penicillium mycelium may be homothallic in nature or heterothallic in nature. Suppose the sex organs are developed from the same mycelium means those species are called homothallic species. Suppose both the sex organs are developed on different mycelium means those are called heterothallic species. So, like this, two types of Penicillium species were identified in this genus Penicillium. So, the sex organs, male and female sex organs, which are actually involving in the sexual reproduction process, how these are produced from the mycelium, how they will develop, what kind of activity they will show during the sexual reproduction process, we will discuss now. So the male reproductive structure in Penicillium is called as antheridium and the female reproductive structure is called as ascogonium. So both male and female reproductive structures, antheridium and ascogonium, will develop in this Penicillium vermiculatum species. So male one is called as antheridium and female one is called as ascogonium. These two sex organs will develop from the mycelium of Penicillium. So how the mycelium is present in Penicillium? Mycelium is a well-developed mycelium. It is a branched mycelium. That means it shows branches. So from this well-developed branched mycelium, one of the vegetative cell which shows lateral growth. One of the vegetative cell shows lateral growth. So from any one of the vegetative cell of the mycelium, lateral outgrowth will develop. So that lateral outgrowth further elongated and increases in length and develops into a narrow elongated tube-like structure that is called as ascogonium. So in this sexual reproduction process, which sex organ is developed first? Female sex organ, that is ascogonium, will develop first. How it will develop? From which portion of the mycelium it will develop means from the well-developed branched septate mycelium. Any vegetative cell will grow laterally. So any vegetative cell which shows lateral growth. So as a result of this lateral growth, one elongated narrow structure which is a tube-like structure will develop. That structure is called as ascogonium. That structure is called as ascogonium. In this image you can see how young ascogonium is developed from the mycelium. You can see here. So like that, from any vegetative cell of the mycelium, one outgrowth will develop. Laterally it will develop. That is called young ascogonium. Further, it shows increases in its length. That means this young ascogonium will grow in its length, so it will elongated. Elongated means long. Lengthwise, it shows increase. So long structure. This ascogonium will develop as a long tube-like structure. This ascogonium will develop. This ascogonium comprises many nuclei. That means inside this ascogonium you will find cytoplasm and nuclei. Many nuclei are present. Actually this ascogonium is a septate in condition. That means there is no cross wall formation or septa formation inside this ascogonium. So ascogonium is a septate in condition. There is no septa formation in this ascogonium. Many nuclei are present. So the presence of multinucleate condition you can see in this ascogonium. So multinucleated cytoplasm is present inside this ascogonium. There is no septa formation initial. Like that, first ascogonium will develop. Later, it will increases in its length and it becomes a tubular elongated structure. Okay, like that, ascogonium, the female reproductive structure will develop. Later, the male reproductive structure antheridium development will take place. Here you can see image C. Okay, first ascogonium, female reproductive structure will develop, then male reproductive structure development will take place. Male reproductive structure is known as antheridium. So how it will develop? Just beside this ascogonium, from another vegetative cell of the mycelium, this antheridium will develop. Okay, just from the same mycelium, just beside to this ascogonium, one vegetative cell develops into antheridium. So initially this antheridium is short in size and columnar in shape. So such kind of antheridium antheridium will develop at the base of the ascogonium. Further, it is also increases in its length and it coils around the ascogonium. You can see, if you observe the image C and D, you'll understand how this antheridium, male initial, is coiling this female reproductive structure. You can see clearly in this image C and image D. So, first antheridial development will initiated. After initiation of male reproductive structure antheridium, this antheridium grows well and which coils around this ascogonium. So, it coils around this ascogonium. Like that, it will show growth. Finally, whenever it reaches maturity, then what will happen? The tip of the antheridium will touches the ascogonium at one point. What will happen here? Important step will take place here. What is that? The tip of the antheridium, that means male sex organ, will touches the female sex organ ascogonium. At that point of contact, the cell walls will be ruptured or dissolved. What will happen? At the point of contact, the cell walls of antheridium and the cell wall of ascogonium, both will be dissolved or ruptured. So as a result, what will happen? The cytoplasmic content of antheridium will mixes with the ascogonial cytoplasm. But the nucleus of the antheridium will not enter into this ascogonium. Nuclear fusion does not take place here. Just cytoplasmic contents mixing only takes place here. That kind of fusion is called as plasmogamy. That kind of fusion is called as a plasmogamy. Just here, antheridial cytoplasm mixes with ascogonial cytoplasm. Plasmogamy will take place. But the antheridial nucleus, still it is present inside the antheridium only. It does not involve in this fusion process. It will never mixes with the ascogonium nucleus or ascogonium nuclei. Only cytoplasms will mix here. Afterwards, after mixing of this cytoplasm, what will happen? The nuclei, many nuclei are there in ascogonium, no? These nuclei are organized in the form of pairs. How these many nuclei are organized in the ascogonium part? Many nuclei, whatever it is present inside this ascogonium, they are arranged in the form of pairs. That means two nuclei come together and arranged in the form of pairs. So, like that, these many nuclei which are present inside this ascogonium will arrange in pairs. That condition, that pairing of nuclei called as dicaryons. That pairing of two nuclei is called as dicaryotization. Okay, they will arrange in the form of pairs. These pairs called as dicaryons. Dicaryons. Caryon means nucleus. Two caryons are there. That is why those are called dicaryons. So, nuclei which are arranged in the form of pairs. So, here the nuclei, whatever it is present inside this ascogonium, which are arranged in the form of dicaryons, that kind of reproduction is called as autogamy. This is one important feature which was identified in penicillium. What kind of reproduction? What kind of fusion this is? Autogamy type of fusion. That means here the nuclei of ascogonium only involves in this process. They are arranged in the form of pairs. That is why this process is called as autogamy. That is why this process is called as autogamy. Here the male nucleus is not involving in fusion of nuclei. Okay, only female nuclei are organized or arranged in the form of pairs. That is in the form of dicaryons, they are organized. That is why this is called autogamy. Important characteristic feature. This was reported only in this penicillium. So like that, dikaryotes formation will take place in ascogonium. Later, what will happen? Now, after dikaryotes formation, septa formation will take place in this ascogonium. That means cross walls formation will take place inside this ascogonium. So what will happen? Earlier, it was aseptate in condition. In the initial stages, ascogonium is aseptate. There is no septa formation inside this ascogonium. But once nuclei are arranged in the form of pairs, then septa formation will take place. So in each chamber, once a septa formation completed means that ascogonium becomes cellularized. Once septa formation completed means what will happen? The total chamber will become cellularized. That means small chambers kind of structure will develop. So you can see cellularization in this ascogonium. So in each chamber, 2 nuclei are present. In each chamber, how many nuclei are present? 2 nuclei are present. See, if you observe diagram F, you can understand this septa formation and this cellularized ascogonium. In each chamber, 2 nuclei are present. Till this septa formation, ascogonium is haploid in condition. Haploid means the nuclei are haploid in condition. They are not fused with each other. Many nuclei are present, but these nuclei are not involving in the nuclear fusion process. They are not fused with other nuclei. So, still septa formation. This ascogonium shows a haploid phase. Once septa formation completed, in each chamber, 2 nuclei are there, no? These 2 nuclei fuses with each other. So nuclear fusion will take place. Nuclear fusion will take place. So diploid nucleus formation will take place here. What will happen? Diploid nucleus formation will take place. So in each chamber, 2 nuclei are present. Both are haploid in condition in the beginning. Then they 2 involves in the fusion process. Those 2 involves in the fusion process. As a result of this nuclear fusion, diploid nucleus will develop. So this ascogonium shows many chambers. In each chamber, 2 nuclei are present. Both nuclei are involved in the fusion. As a result of fusion of 2 haploid nuclei, results into formation of one diploid nucleus. Results into formation of one diploid nucleus. Like that, diploid nucleus will develop in each and every chamber. Still, the antheridium retains its nucleus with it only. The nucleus of antheridium will not enter into this ascogonium. It will not fuse with any nucleus of this ascogonium. Still, antheridium retains its nucleus itself only. So, diploid nucleus formation completed. At that time, from each chamber, from this ascogonium, some filamentous structures will develop. From this ascogonium, some filamentous structures will develop. Those filaments are called as ascogenous filaments. Some filamentous structures will develop from this ascogonium. Those filamentous structures are called as ascogenous filaments. Like that, many filaments will form. These filaments will show growth. So these ascogenous filaments also increases in its length. So they will become elongated. Ascogenous filaments also increases in its length. So what will happen? They will become elongated. Ascogenous filaments will elongated in length. Okay. So later, finally what will happen? The tip of the ascogenous filament will be arc-like in shape. Arc, half moon shape. The tip of the ascogenous filament attains one half moon shape, that is called crozier. that will functions as ascus mother cell. The tip of the ascogenous filament which attains a arc shape which is called as crozier. That tip region will functions as ascus mother cell. That tip region will functions as ascus mother cell. Inside this ascus mother cell, one diploid nucleus is there. Inside this ascus mother cell, what is present? One diploid nucleus is there. So, this tip region, which is called as crozier, which is functioned as ascus mother cell. This ascus mother cell possesses one diploid nucleus. That diploid nucleus now will undergo meiotic division or reduction division. So the diploid nucleus containing ascus mother cell will undergo meiotic division or reduction division. So as a result of this meiotic division or reduction division, 4 haploid nuclei will form. These 4 haploid nuclei further undergoes mitotic division. So totally 8 haploid nuclei will form inside this ascus mother cell. Understand? From the tip of the ascogenous filament, one arc-shaped structure crozier will develop that will function as ascus mother cell. That ascus mother cell possesses one diploid nucleus. That diploid nucleus divides by meiosis first, then it will undergo mitosis. So as a result of this meiotic division, 4 haploid nuclei will form. These 4 haploid nuclei undergo mitotic division. So as a result, finally 8 nuclei will form. All these nuclei are haploid in condition. All these nuclei are haploid in condition. Further, each and every nuclei, 8 nuclei are formed now. All these 8 nuclei gets some amount of cytoplasm from the ascus mother cell cytoplasm, and they will become modified into ascospores. Okay, 8 haploid nuclei formed now. Each and every nuclei gets some amount of cytoplasm. So after getting this cytoplasm, the nucleus along with the cytoplasm, it will modify into one spore. That spore is called as ascospore. That spore is called as ascospore. So like this, from one ascus mother cell, how many ascospores will form? 8 ascospores will form. Each ascospore consists some amount of cytoplasm and one haploid nucleus. Like that, from one ascus mother cell, 8 ascospores will form. So like that, ascospores will be produced inside the ascus mother cells. Okay. So like this, ascus and ascospores formation will take place. At that time, what will happen means another important process will take place here. That is, already you have learned about the antheridian development and ascogonian development in this sexual reproduction process. How these two are developed in such a fashion, from the main mycelium, some vegetative filaments also started developing from the base of this ascogonium and antheridium. Some vegetative filaments will develop from the base of ascogonium as well as antheridium. From which portion these are developed? These are also developed from the mycelium, fungal mycelium only. This vegetative filaments development also will take place. So these vegetative filaments mixes with ascogenous filaments. So the vegetative filaments which are developing from the fungal mycelium mixes with ascogenous filaments, and they will form one fruiting body. These vegetative filaments along with ascogenous filaments, one fruiting body or one ball-shaped structure will develop. That fruiting body is called as ascocarp. That fruiting body is called as ascocarp. Actually, in this fungi, different types of fruiting body formation we will see. Here in this penicillium, what type of fruiting body formation will takes place means the fruiting body is formed without an opening. It is completely closed structure. That kind of completely closed fruiting body will develop, which is commonly called as ascocarp. Here, different types of ascocarp formation will takes place in fungal species. Here, the fruiting body which was developed in Penicillium vermiculatum is a closed fruiting body. That means there is no opening to this fruiting body. There is no ostiole. There is no opening means one structure which is called pore-like structure, that is called ostiole. That kind of ostiole or opening is not there in this fruiting body. So such type of fruiting body is called as cleistotecium. What type of fruiting body will develop in this Penicillium vermiculatum? Cleistotecium type of fruiting body formation will take place. That means that this cleistotecium is a fruiting body, that is ascocarp, that does not possess any opening or ostiole. So here it is formed with ascogenous filaments and vegetative filaments of the fungal mycelium. Like that, this fruiting body will develop. So inside this fruiting body, ascogenous filaments are there. From this ascogenous filaments only ascus will form. Inside that ascus, this ascospores are produced. So like this, sexual reproduction process will completed. Okay. So here, due to autogamy, dikaryotic condition will arise. These dichorions further undergoes fusion process. So diploid nucleus formation will take place. This diploid nucleus undergoes meiotic division. Further it undergoes mitotic division. So as a result, from each ascus mother cell, 8 haploid nuclei will form. This 8 haploid nuclei gets some amount of cytoplasm and they will become ascospores. They will modify into ascospores. So 8 ascospores are produced from each ascus. Okay. So these ascus are produced at the tip of the ascogenous filament. So these ascospores are present inside the ascus. Ascus is present at the tip of the ascogenous filament. So these ascogenous filaments are present inside this cleistotecium fruiting body. So like this, fruiting body development also completed. So here, this ascospores reaches maturity. Whenever the ascospores reaches maturity, then the ascus walls will be degraded or those will be degenerated. Ascus is protected by one protective wall or protective sheath. That protective wall layers of ascus will be degenerated whenever ascospores are matured. Then these ascospores are liberated from the ascus. So once the ascus wall is ruptured, these ascospores are released, means they are released into the cleistotecium. Then they have to release from the cleistotecium. Cleistotecium is fruiting body. So later the cleistotecial wall layers, that means periderm. Cleistotecium is covered with one protective covering, that is called periderm. Cleistotecium is a fruiting body that is covered with one protective covering, that is called periderm. So this periderm also ruptured. So due to the rupture of this periderm, these ascospores are released from this fruiting body. These ascospores are released from this fruiting body. So like this, sexual reproduction will take place in this fungus Penicillium vermiculatum. This sexual stage is called as perfect stage. This is named as Talaromyces vermiculatus. The sexual stage of Penicillium vermiculatum is named as Talaromyces vermiculatus. So like that, ascospores are produced as a result of this sexual reproduction? These ascospores are matured, then they will be liberated from the ascus. Then they have to liberate or they have to release from the fruiting body cleistodecium. That was also completed by the degeneration of periderm layers. Periderm means protective covering of cleistodecium. So finally, these ascospores are released from the cleistodecium. Whenever these ascospores are released from the cleistodecium like this, see, this is about a cleistodecium. This outer layer periderm or peridium, which is ruptured on maturation of ascospores. What will happen? This periderm or peridium will be removed. So as a result, these spores, ascospores, are released from this fruiting body cleistodecium. These are released from this fruiting body cleistodecium. So what is the fate of this ascospores now? These ascospores, whatever it is released from the fruiting body cleistodecium, they will fall on different substratums. Whenever they will get favorable conditions, unless and until they will get favorable conditions, they will undergo resting phase. So once they will get favorable conditions for their growth, these ascospores germinates and produces germ tube. These ascospores germinate and produces, see, this is the structure of a cleistodecium. This is the structure of a cleistodecium. By the rupture of this outer wall, peridium, these ascospores are released. Once they reaches onto the surface of suitable substratum and the conditions are favorable for their growth, each ascospore germinates into germ tube and it produces one fungal mycelium. Like that, ascospores will germinate and grows into fungal mycelium. Like that, ascospores will be released from the fruiting body cleistodecium and they start germination and they'll germinate and give rise to fungal mycelium. Like this, ascospores will germinate and gives rise to fungal mycelium. This is about sexual reproduction in Penicillium vermiculatum species. So the life cycle, if you observe in Penicillium, which phase is dominant means haploid phase is dominant because this fungus reproduced asexually. By asexual means of reproduction, haploid conidia or conidiospores are produced. So the haploid phase is dominant in Penicillium life cycle. Diploid phase is confined to only this dikaryotic condition stage only. That means inside the ascogenous filaments or inside the ascogonium, two nuclei fused, so diploid nucleus formation will take place. So the diploid stage or diploid state is confined to only short period. So diploid stage is a short-lived phase. Remaining life cycle, the haploid phase is dominant. So only for a short period of time it is in diploid state. Remaining period it is in haploid state. So the life cycle of Penicillium is described as a haplontic life cycle. Okay, during sexual reproduction, only in the species which are exhibiting, which are showing sexual reproduction process, in those only, that too only for a short period of time, this diploid condition will appear. Remaining time it is haploid in state. So the life cycle of Penicillium is haploid. So the life cycle is described as haplontic life cycle. What type of life cycle this Penicillium will show? Haplontic life cycle. This Penicillium will show. This is about sexual reproduction in Penicillium. Next, economic importance of Penicillium also very important. So that one also I want to explain. You may get some important bits from this economic importance area. So how this penicillium is important economically, we will discuss now. Just briefly I will explain you. Actually, the species of penicillium are having great economic value. These are not only having beneficial effects, these are showing some harmful effects also. So we will discuss about harmful effects of penicillium species as well as beneficial effects of penicillium species. In what way these penicillium species are causes damages, we will discuss under this harmful effects category. Actually, moist conditions or damp conditions favors the growth of penicillium species. So, if the environment is having moisture, moist conditions are present means this fungus penicillium will grow fastly. So, moist conditions favors the growth of penicillium species. In such kind of moist conditions or damp conditions, this penicillium species attacks variety of substratums. On what kind of substratums these will attack? In the introduction part yesterday I have explained. These penicillium species attack on furniture, they will attack wood, they will attack leather products, they will spoil fabrics, they will spoil paper also. So, whenever moist conditions are available, in such kind of moist conditions, this penicillium species will attack fabrics, furniture, wooden material or wooden articles, leather items. All these are attacked by this penicillium species and they will spoil this all materials. So, this is one of the harmful effect of penicillium species. Next, one particular species that specifically attacks wooden articles or wooden items that will give yellow color, pale yellow color to that wooden articles. So the quality of wood and wooden articles will be decreased gradually. That species name is Penicillium divaricatum. What species spoils this wooden articles? Penicillium divaricatum species gives yellow color, pale yellow color to the wood items. So the quality of that particular wood items will be decreased greatly. So that particular species is Penicillium divaricatum. Next, whenever conditions are wet, so in such kind of wet conditions, this Penicillium species will attack foodstuffs. So they will spoil the food items. For example, breads, cheeses, butter, jams, jellies, pickles, all these are spoiled by the Penicillium species growth. So they will attack on foodstuffs and they will spoil these foodstuffs. But these foodstuffs are not poisonous by this Penicillium species growth. They will reduce the quality of the foodstuffs, but these foodstuffs after the Penicillium species infection, they will not become poisonous. So they are not poisonous, but quality of that food items will be decreased greatly. So like that, they will spoil variety of food items. So moist conditions will favor this Penicillium species growth on food items. So we have to preserve these foodstuffs at dry conditions. Next, some of the particular Penicillium species will spoil and damages the citrus varieties. Citrus fruits, all of you know, lemons, oranges, etc. These citrus fruits are spoiled by the growth of Penicillium digitatum species. This species will be, this species will causes green molds on the citrus species fruits. So This citrus species fruits are damaged greatly by this Penicillium digitatum species. Next, another species of Penicillium, that is Penicillium italicum, that spoils oranges, and this Penicillium italicum species develops on this oranges and lemons as blue molds. Digitatum will develop as a green mold on citrus fruits. Penicillium italicum species develops as a blue mold on the citrus varieties. Another species that is also comes under this genus Penicillium. That species especially attacks the apples. Whenever apples are stored in that storage period, apples are damaged greatly by Penicillium expansum species. So this particular species causes great losses to the apple cultivars. So during storage period and during transportation period, apples are spoiled and damaged greatly by this Penicillium expansum species. Another species, that is Penicillium purpurosinum. What is that? Penicillium purpurosinum species. That will cause some spots on the paper and books and reduces the quality of papers and books. That particular species name is Penicillium purpurosinum. This Penicillium purpurosinum attacks on the books and paper material. So they will reduce the quality of these books and papers. It causes great losses in paper industries. This is about the harmful effects of Penicillium species. Next, we will discuss about beneficial effects of Penicillium species. Till now, we have discussed about how these Penicillium species are causes losses to farmers, some industries. We have discussed leather industries, paper industries, etc. We have discussed food industries, how these are affected by this Penicillium species. We have discussed. Now we are going to discuss about useful aspects of this Penicillium species. Penicillium species not only causes damages and losses to fruits, vegetables, foodstuffs, papers, leather items, wooden articles, etc. Some of the Penicillium species are useful to human beings in a great way. How these are useful to human beings means here some of the Penicillium species which are used extensively in the industries for the production of important medicines. For example, variety of antibiotics produced from this Penicillium species. I hope all of you know about wonder drug. What is the common name of wonder drug? Penicillin is commonly called as wonder drug. Why it is named as wonder drug? Because during Second World War time, this drug saved lakhs of soldiers' lives. That is why this antibiotic penicillin is known as wonder drug. Who discovered this wonder drug? Alexander Fleming discovered this wonder drug. He studied about the medicinal properties of this penicillin along with two other scientists of Oxford University. Those scientists' names are Professor Flory and Professor Chain. Along with these two scientists, Alexander Fleming studied about the medicinal properties of penicillin. So for their contribution, Alexander Fleming and Chain received a Nobel Prize. So this drug saved lakhs of soldiers' lives during Second World War time. That is why this penicillin is commonly called as wonder drug. So that wonder drug penicillin was obtained from the genus Penicillium and the species Penicillium chrysogenum. Earlier this chrysogenum is called as Penicillium notatum. So that much powerful drug was extracted from this Penicillium species only. Next, another antibiotic that is also having great importance, that is griseofulvin. This is another drug. This is also antibiotic. That antibiotic name is griseofulvin, which is extracted from the species Penicillium griseofulvum. This drug or this antibiotic is used to treat ringworm. This antibiotic is used to treat ringworm in human beings as well as animals. Not only this drug used to treat ringworm in human beings and animals, it is also used to treat some fungal diseases of plants. This griseofulvin antibiotic is used to treat some fungal diseases of plants. This is about another important antibiotic which is also obtained from the genus Penicillium only. That is species griseofulvum species. Penicillium griseofulvum species is used to produce griseofulvin antibiotic. So these antibiotics are produced on large scale in the industries by fermentation process by using this Penicillium species only. This Penicillium species not only used to produce variety of antibiotics, these are also used to produce some commercially important organic acids like gluconic acid, citric acid, formic acid, like that variety of organic acids, for example oxalic acid, fumaric acid, gluconic acid, gallic acid, such kind of commercially important organic acids also produced by using Penicillium species. Most commonly used Penicillium species to produce these organic acids are Penicillium purpurosinum, Penicillium digitatum, Penicillium glaucum. Penicillium purpurosinum, Penicillium digitatum, Penicillium glaucum. These species are extensively used in the industries for a large scale production of commercially important organic acids. These Penicillium species not only used for the production of industrially important organic acids but also used for the production of commercially important enzymes. For example, cellulases, proteases, pectinases, like that different types of enzymes also produced on large scale by fermentation process. For that enzymes production also this Penicillium species are used. By using this Penicillium species by fermentation process, different types of enzymes are produced on large scale. For that purpose also these Penicillium species are used. So Penicillium species not only used for the production of antibiotics and organic acids. These are used for the production of enzymes. Okay. Next, in India, one government organization that is producing this penicillin. That organization name is Hindustan Antibiotics Limited, which was located at Pimpri, Pune. This is producing this penicillin. So this penicillin is used to treat several bacterial infections. How this penicillin will work? Actually, this penicillin will work on bacterial cell walls. How it will work on bacteria, how it inhibits the growth of bacteria means it stimulates the production of some enzymes which prevents or inhibits the cell wall formation in bacteria. In such a manner, these bacterial diseases and bacterial infections are treated by using this drug penicillin. Understand how this penicillin is used to treat bacterial infections and bacterial diseases. Here, this penicillin antibiotic inhibits the cell wall formation in disease causing bacteria. So how this cell wall formation is blocked in this bacteria? The enzymes which are able to block cell wall formation in bacteria, those enzymes production will be stimulated by this penicillin antibiotic. In such a manner, this penicillin inhibits the growth of disease causing bacteria. In such a manner, this is used to treat various bacterial diseases. Penicillin actually acts on some important bacteria which are very harmful and serious disease causing bacteria. Those are Streptococcus bacteria, Pneumococcus bacteria, Meningococcus bacteria, and Staphylococcus bacteria. Penicillin antibiotic effectively works on Streptococcus strains, Staphylococcus strains, Pneumococcus strains, and Meningococcus strains. This drug can prevent the diseases caused by all these four strains of bacteria. So like this, penicillin will works. This is about economic importance of penicillium. Here I forgot to mention one important point. With that I'll conclude this class. That is, here penicillium species not only used to produce enzymes, organic acids, antibiotics, etc. Some species of penicillium are used to prepare some tasty desserts or cheeses. Especially Penicillium camembert species is used extensively in the production of camembert cheese. Another species of Penicillium, that is Penicillium roqueforti, that is used for the production of roquefort cheese. So these two species of Penicillium, that is Penicillium camembert and Penicillium roqueforti, gives some special flavor to that particular cheese, and they will give some soft texture to that particular cheese. They are also giving color to that particular cheeses. So because of these reasons, these two species are extensively used for the production of camembert cheese and roquefort cheese. These two cheeses are prepared by using Penicillium camembert species and Penicillium roquefort species. This is about beneficial effects of penicillium species. So with this, I want to conclude today's class. I hope all of you understand today's lecture. Thank you, sir.
Okay, madam. Really is a nice presentation, madam. Because of very busy schedule, you are spare your valuable time for our young aspirants. Definitely, they will get good score in CPGET 2023, madam. Your class is very live mode, very effective manner. We are all enjoy with all the useful information regarding of this penicillin and also the useful effects of penicillin and the penicillin industry, antibiotic activities, enzyme activities, and also used for the organic acids. So, and also the discovery of the penicillin is also very useful for all competitive examinations of our plant sciences. Really wonderful presentation, madam. Thank you very much. So tomorrow we will meet same time. Thank you, madam.
Okay, sir. Thank you, sir.